Hi everyone, my name is Marina Tagliaferro. I'm presenting this work, which was made in collaboration with these persons, Verónica Villanueva from Idioma, Laura Wolinsky, and Claudia Boy. We are from Cali, which is located in Australia. Before I start, I would like to thank uh, the Society for Freshwater Science for helping me to participate with the grant for early career researcher from Latin America. So I was explaining this habitat and showing this picture is from Australia, which is located in southern South America and in the Andes, indeed. And these kind of habitats and the environments are very close related to the terrestrial and the aquatic. So they are very close. Indeed, streams here are heterotrophic in general, and there are natural relationships that we already know about the macroinvertebrates and the life cycle, which requires some stages in the water. But then with the immersions of these organisms, they go to terrestrial environment and then they return to the water. And also there are many links between these two habitats. Here you can see that we have debris, we have wood, and also the main source for these organisms in this area are leaves that come from the forest, which is not a palus forest. So these kind of forests are mainly related to the Andes, to the southern part of the Andes, in Patagonia mainly, from Chile, Argentina, but also there are other species present in New Zealand. So I'm mainly a freshwater ecologist, so I was working in freshwater habitats, and when making some studies, I found that I have one type of forest with a low presence of galls and another forest with a large uh, frequency of these galls. So this habitat where we work and all the habitats along the Patagonia are experiencing these relationships with other terrestrial organisms that are parasitic and that are affecting the natural forest with these galls that are really big ones and affecting like the 30 to the 50% of the area of the leaves. So when I was seeing this, I started asking myself, uh, are these galls affecting the quality of the leaves? And what about the quality of the organisms that leave these leaves as resource? So the aim of this study was to compare the consumption of these leaves by these two organisms. One is caddisfly, the other one a mosquito, and I'm so sorry for my English. And I was also considering the effects on the nutrient. So we have different predictions. I will show only a few of them. One is that these galls might have higher nutrients associated, mainly because the leaf activation is earlier during the year. Um, Before the autumn came, these galls go to the floor and then to the water. Then I was expecting a different relationship in the quality depending on the functional feeding groups because they use different source, but they use it in a different way. So I was also expecting some differences in the spectrometry. The methodology we used it was, well, we did some a field sample, something. We took samples and analyzed leaves, invertebrates, and water. And then we make lab experiments, and for that we needed to go to the field also to take the leaves and condition the leaves. Uh, and we made this experiment with these two species for 30 days at the temperature that is natural in our stream, that is 5 degrees Celsius. Um, okay, considering the habitat, the water radiation, and all the needs for the invertebrate, and we gave them these two quality of food. So we measure carbon, nitrogen, and phosphorus from the leaves, the water, and the organisms. Also, ergosterol to evaluate the biomass, to estimate the biomass of the fungi, and also the consumption and the weight. And we analyze this with an ANOVA. So the first thing I want to be clear is that even we have one referent forest and we have a infected forest, both forests have leaves that are not affected with galls, okay? And the forest which is infected has the two types of leaves, with galls and without them. So this was interesting for me. What we found, the first thing was to analyze if they were different, and the lead from the reference and from the infected forest, they were similar in carbon and nitrogen and the relationship. But when we consider the the reference to the Gulf um, leaves, we found they have a higher nitrogen, similar phosphorus, but the relationship of carbon phosphorus was higher in this infected forest. They have a higher ergosterol, also higher nitrogen and phosphorus, and lower carbon nitrogen rate. So considering this, we evaluate. I need to move myself again. Um, and with this, what we found was that the consumption was different between the two resources, but the relation was almost the same. It was we had high consumption of the gall leaves and a lower of the control mass. And this relationship was the same if we related to the way that the organisms are not. So then we analyze the stoichiometry of we evaluate the consumer stoichiometry, and we found that people experienced had a lower nitrogen content than the caliculi, and then the phosphorus content was higher. While the radial carbon nitrogen did not differ, the carbon phosphorus did differ, and also differ between gulf and ungulf treatments, and also the species. And the relationship was not the same. It was contrary. When we evaluated the relationship of the lead here, nitrogen phosphorus of the food and the organism, what we found is that people naturally tend to be balanced. This line in the middle, dotted line here is like the balance, the 1-1 relationship. So in relation to reference food, it is balanced and tend to be unbalanced when they are fed on balanced um, plates. On the other hand, the caddisflies are highly unbalanced and tend to be more balanced when they fed on gathered leaves. Um, so our conclusions now are moving. Um, our conclusions here are that these healths are affecting the stoichiometry and the nutrient content, so they are affecting the quality. And they are also affecting other things. For example, the moment that they go to the soil, to the forest, they they have a higher nutrient content, but they are also heavier. So they stand in pools together. So we have a high quantity with a higher quality of nutrients. Also, they are associated with a higher fungi biomass, and also these qualities are affecting uh, the conditions of macroinvertebrates that are feeding on these resources, which are new resources. Um, and this relationship will be affected also by the type of functional feeding groups of the organisms. One is more effective than the other one, and also they depend on their natural condition. So I would like to thank again the Society for Freshwater Science, the finance from Subsides, my institution. I would like to thank the people that helped me in the field. And I would like really to thank Alonso Ramirez for helping people from Latin America, and Pablo, which was a referent for early career to assist to this meeting. Thanks all.